Okay, let's start. Hello, welcome. Today's guest lecture is on a synthetic biology approach of RNA-guided precision targeting of cancer cells. My name is Chami Samarasekar from the ANU College of Health and Medicine, and I'm joining in from the beautiful campus in Canberra. Before we start, I wish to acknowledge the Ngunnawal and Ngambri people, the traditional custodians of the land I am at today presenting, and recognize any other people or families with connection to the land of Australian Capital Territory and region. I wish to acknowledge and respect their continuing culture and the contribution they make to the lives of this beautiful city and the region I live in. I also know we are joined by people from around the world, so I would like to welcome all the Aboriginal and Torres Strait Islanders and Indigenous people who may be attending today's webinar from across the country and anywhere in the world. So let me now introduce you to our amazing presenters today. I'm sure you have all read their bios, so I'll try to summarize as much as I can. First, we have Associate Professor Thomas Fisher from the John Curtin School of Medical Research. We call it JCSMR for short. He completed his PhD at the University of Heidelberg on the nuclear export in mRNA and its coupling of transcription. After that, he was awarded a postdoctoral fellowship in epigenetics and chromatin biology at the National Cancer Center in the USA. He returned to Germany to establish his own independent research group at the University of Heidelberg Biochemistry Center, and he has recently moved his laboratory to Canberra to join the Division of Genomic Science at JCSMR. We also have Associate Professor Marianne Berg joining us. Professor Berg completed PhD in immunology at the University of Cambridge. Her clinical specialty training in anatomical pathology inspired her to apply her expertise to tackle the problem of immune evasion in cancer. Supported by prestigious Cancer Research UK Clinician Scientist Fellowship, she established a highly productive research program in Cambridge and subsequently at the Peter MacCallum Cancer Centre here in Australia, making fundamental biological discoveries that hold great promise for development of novel cancer immunotherapies. In 2017, her team identified previously uncharacteristic protein CMTM6, hope I'm saying that right, to be a binding partner and regulator of PD-L1 in cancer cells, a landmark discovery that has opened a new field of research and led to establishment of a drug discovery program in Australia and worldwide to develop immunotherapies targeting the CMTM6 PD-L1 interaction. Her recent work and discoveries have uncovered new opportunities for targeted use of epigenetic inhibitors to improve anti-tumor immune responses in a number of aggressive, difficult to target malignancies such as neuroblastoma and lung cancer. So I welcome both professors to today's lecture, and I'll hand it over to you.
Okay, so thank you for the introduction, and thank you everyone for joining today. So I'm delighted to present what is a collaborative project. So I will be presenting for the first part of the seminar, and then joined by Thomas Fisher, who will present more details of the molecular tool that we're developing. So today we're talking about a synthetic biology approach for RNA-guided precision targeting of cancer cells. So for the current status of cancer therapies, I'm going to mainly focus on current treatments for lung cancer, which is the main area in which my lab is working. But really the same principles apply to many different forms of cancer. So if patients present with a localized tumor that's surgically resectable, then that's the preferred treatment option, which often, if this is caught early, can be curative. However, unfortunately, for lung cancer, most patients present with advanced disease. That's disease that has already metastasized and spread to other parts of the body. In that setting, there's a range of different systemic therapies that can be used, and these can be used to prolong life, but unfortunately are often not curative. So the traditional mainstay of treatment has been chemotherapy and radiotherapy, and then more recently we've had the introduction of targeted therapies. So these target specific mutations in the tumor cells which drive the tumor cell proliferation. So these are frequently in oncogenic tyrosine kinases such as EGFR, ALK, or KRAS, and specific inhibitors have been developed to inhibit these pathways. Now, in the context of these systemic therapies, and here I'll use the example of chemotherapy, these therapies target fundamental essential cellular processes. So the basis on which they're effective for cancer is that they target processes which are disrupted in cancer cells. And so that means that the tumor cells will have a more selective dependency on these pathways. So for example, because tumor cells are proliferating more rapidly or have impairment in their DNA repair pathways. However, because these chemotherapies are also targeting fundamental cellular processes, they will also target normal cells. So this means that at higher doses, these therapies will have dose-limiting toxicities and cause severe side effects. So this leads to the concept of a therapeutic window in which there will be an effective dose which may be selectively effective in targeting the tumor cells without causing too severe side effects. However, unfortunately, ultimately, the tumor cells will acquire resistance to these therapies, which means that these therapies largely won't effectively completely eradicate the tumor. So this is the case for chemotherapy, but actually is the case for all other current therapies that we use. So the same applies to radiotherapies. And even for targeted therapies, the therapeutic window is often larger, but ultimately these are still targeting normal cellular pathways and so are going to affect the non-tumor cells. Now, one of the more recent developments and what is the main focus of the research in my lab is in immunotherapies, and specifically in immune checkpoint inhibitor therapies, which are now widely used in the clinic for lung cancer. So immune checkpoint inhibitor therapies, and these, the ones most commonly used for lung cancer are antibodies which block the interaction between PD-1 and PD-L1, which is expressed on the tumor cells. These activate a tumor antigen-specific cytotoxic T cell response. So under normal circumstances, CD8 T cells recognize antigens which are processed from intracellular proteins in cells and presented on the surface in complex with MHC class I molecules. Now, CD8 T cells serve as the patrolling cells which allow the immune system to discriminate self from non-self. So in that context, CD8 T cells are able to recognize cells which have become aberrant and are presenting aberrant peptides. So in the context of cancer cells, these abnormal peptides can arise either from mutations or aberrantly expressed proteins. This means that the tumor cells are recognized as foreign and so can be selectively eliminated by the CD8 T cells. However, tumor cells establish mechanisms by which they can evade this CD8 T cell recognition and killing. And one of the most common ways that they do this is by expressing inhibitory ligands on the cell surface, such as PD-L1, which then binds to PD-1, which is an inhibitory receptor on the T cell, which inhibits CD8 T cell activation. So immune checkpoint inhibitor therapies have been developed to block this interaction with antibodies which block the interaction between PD-1 and PD-L1. This leads to CD8 T cell reactivation and tumor cell killing. So these immune checkpoint inhibitor therapies have been incredibly effective in certain forms of cancer. This just shows an example from metastatic melanoma, in which the historical survival was less than 5%. And now with immune checkpoint inhibitor therapies, over half of patients are surviving long-term. However, these therapies still are not completely tumor specific. And the reason for that is that although the CD8 T cells are able to selectively target the cancer cells, the therapies themselves leads to systemic activation of the immune system. And therefore, this can cause systemic inflammation in almost every organ. So the most common limiting side effects are inflammation in the liver, causing a hepatitis, in the lung, causing a pneumonitis, or in the gastrointestinal tract, causing a colitis. And these toxicities have meant that for some patients, they're not able to continue these therapies. In addition to this, many tumor cells also display resistance to immune checkpoint inhibitor therapies, which means that although the melanoma is the tumor which has so far responded the best, in many other tumor types, the response rates have been much lower. So this has led us to the question to try and understand and try and develop an approach which may allow us to more specifically target the cancer cells themselves while avoiding effects on normal healthy tissues. And so with that, I'll hand over to Tamas Fischer, who will tell you about the approach that we are using.
All right, so thanks, Marianne, for introducing the project. And as Marianne mentioned, I will now show a little bit more the molecular details, what is our approach, and then I will hand over back to Marianne to kind of put the picture together. All right, so when I started to think about this problem many years ago already, how could we really have this specificity in cancer treatment, so that we really only specifically targeting cancer cells and not all the other cells, as Marianne mentioned, which cause a lot of side effects and other problems. So we know that cancer cells, so one of the hallmarks of cancer is that these cells have a lot of DNA damage stress, mitotic stress, and they accumulate a lot of mutations. And by a lot, I mean really these are thousands and hundred thousands of mutations. So this kind of eclectic little drawings just want to show you just normal cells at this point, or which then become pre-cancer cells, and this is the genome. And then you can see there are some mutations. So in this case, this red dot and blue dot, those are early mutations which these pre-cancer cells accumulate. And at one point, these pre-cancer cells actually reach the stage where they really start to proliferate and start to grow in the body and accumulate more and more mutations. So you can see all of these different little dots, which means that they accumulate many mutations. Now, if there's a tumor in a patient, and we would take a biopsy, we would take all of these cancer cells, which, as you can see, some has red and blue and orange, while other has blue and red and yellow. So they all have like certain mutations which are common in them, which were kind of early mutations, and then also some which are just picked up during this journey. So the question is, could we target these mutations? So in a way, could we develop a tool which we could find very specific mutations? So for example, here's patient 1. We could take a biopsy from the cancer and sequence it and then determine and find some of the mutations. Also take a biopsy from normal tissues and kind of compare it and really determine which are the mutations which are only in the cancer cells. Of course, this would be not mutations for every cancer. It would be just in this very specific patient. Another patient would have different mutations. So could we use this as a specificity? Now, just to show you that this is indeed, like cancer cells have really many, many mutations. This chart just shows all the different cancers, pancreatic, breast, and so on. So you can see here on top. And each individual dot are here a single patient where they took biopsy and sequence their genome, or actually the exome, so those part of the genome which is expressed in these cells, and then they determined how many mutations, cancer-specific mutations, can they find. And as you can see, this is somatic mutations per megabase of genome. And so the human genome is over 3,000 megabase. So as you see, from the 0.1 up to 100 mutations per megabase, which means in every tumor, or most of the tumors, a patient would have like hundreds to hundreds of thousands of mutations. So there are indeed many of these mutations which could be used, which is very specific in that patient for their cancer cells. Now, I just want to, like Marian spoke about immunotherapies, and I just want to mention that if you think about immunotherapies are actually using these kind of mutations already, because in a normal cell, of course, there's no mutation in the RNA, which is forming a certain protein, and these proteins are non-immunogenic. But in a mutated cell, in a cancer cell, where you can see these little mutations in the DNA, which will be also transcribed, and it will be in the RNA, and this will lead to mutated proteins. And the immunotherapy is in a way based on the idea that these mutated proteins will then present as neoantigens, and so that will be presented in the cell surface, and then kind of our immune system can attack these cells. Now, this is, so immunotherapy is a really important and amazing development, and indeed it gives for the first time real specificity into cancer treatment. However, there are a couple of problems, and Marian mentioned these. One of them is that the prediction from whether which peptide will be immunogenic is quite complicated. It's not really reliable, because this has to be bound by certain molecules which are presenting in the cell surface and so on. So it's not so easy to predict which mutation will actually be immunogenic. Plus, as Marian mentioned, there are many mechanisms where the cancer kind of learns or like evolves to suppress immune response, for example, by suppressing the presentation of these antigens or other ways. So the question was, would it be possible to use directly the mutated RNA? So not focus on the protein parts, but really use these mutated RNAs as a deciding factor whether this is a cancer cell or not. Can we find these mutated RNAs? And if yes, can we eliminate these cells, or can we somehow specifically have a response in these cells? Now, so, and this is what we want to do. So that's our basic kind of objective. So we are developing a molecular device which can, and this device right now, I'm just representing like a little molecular robot, but I will go into more details, which can somehow scan the genome or the transcriptome. Right now here I show DNA, but actually what we are really doing, we scan the transcriptome, so RNAs of a cell for specific markers. And these markers we can program. So this marker would be not the same for patient A and patient B. It would be really patient specific. And so if this marker is found, this specific sequence, then this little device will execute a program, for example, cytotoxicity, or somehow eliminating this cell. Whereas if this device just doesn't find the mutation or the RNA sequence what is searching for, then this device will just self-destruct and disappear from the cell. So we are using synthetic biology to achieve these goals. And I could talk about synthetic biology a lot because it's a very exciting concept. Half of my lab is doing kind of basic biology. And in that case, we are mostly asking how things are working. And I often don't really absolutely care whether it will be very useful or not. I just want to understand how things are working. Now, engineering is a very different idea. In engineering, you are not asking, you are trying to put together something which is doing what you want. And exactly this is synthetic biology. So it's kind of in between biology and engineering, where we use already characterized biological systems, and we are engineering it and putting it together in a way that it does what we want. So we can design and build like small biological systems and functions. In this case, this is what we want to build. So what is the approach? How do we get there? Now, this is almost just a sidetrack, but probably many of you already heard about CRISPR tools, so the various Cas proteins, and probably the most well-characterized or probably heard the most about the Cas9 protein, which can find very specific sequences in the genome using a guide RNA. And this guide RNA you can change and you can specify the sequence. And according to the sequence what this guide RNA is, you program, it will find that very specific sequence in the genome. And actually, the Cas9 will cleave this DNA. Now, there are other Cas tools which were developed. For example, Cas13. In this case, again, you have a guide RNA, but it's not looking for DNA sequences, but it's looking for RNA sequences. So it can find these very specific RNA sequences and indeed cleave this RNA. Now, using these tools, synthetic biologists were thinking, and including us and other groups, could we use this somehow to find specific mutations, but not just cleave this RNA or DNA, but do something else? So kind of make an inactive Cas protein, which is now not cleaving the guide RNA, but put certain effectors to it. And this effector can do various things, for example, induce cell death. And I will not go further in this. We have a project which is working on this, and indeed it works somewhat. However, there are some major problems. One of the problems is that all these Cas proteins are large proteins, which really complicates the delivery to tumor cells. Plus, these Cas proteins are also immunogenic, so it would go to every cell, not just the tumor cell, but even in a normal cell, it would cause an immune response. So it's not really ideal for this purpose. And we realized that actually, to be honest, the job is done by the guide RNA. So the guide RNA itself, which is finding the sequence what we are looking for, the Cas protein is just a cargo, which is kind of following the guide RNA. So we were starting to wonder whether could we just build up a system which is kind of leaving out the whole protein from the picture and only focusing on the RNA part, on the guide RNA part. And indeed, I'm an RNA biologist, and I love RNA because many reasons. But one of the reasons which I try to present in this slide, RNA has really two faces. It can speak the nucleic acid language, so the DNA language or the RNA language, because the RNA can base pair with the DNA or with another RNA. So the RNA really just understands nucleic acid language. The base pairing is just super easy. Also, that's why we are using it as a guide RNA. A guide RNA will find by base pairing the corresponding sequence. However, RNA has also another phase. RNA can make very complex structures, very complex and exciting structures, just like proteins. Also, proteins are normally effectors because they can make very complex structures, but RNA can do the same. So, and this is what we call ribozymes. So in a way, if we are using an RNA, we could put together these two functions. We can put together a guide RNA and immediately attach to it a ribozyme. And this is, so this was the basic concept when we started to work on this. And we started to focus on these group 1 introns, which are cell-splicing ribozymes. So what does it mean, cell-splicing ribozyme? And I'm sorry if I go, I'm not sure exactly what the audience level of understanding what is splicing, so I just quickly explain. So in messenger RNAs have exons and introns in general in eukaryotes. So that means that the message is always coded on the exons, and the introns are non-coding sequences. So if I just focus on this, as you see in our DNA, the messages are coded on these exons, and interrupted by these introns. And when the DNA will be like transcribed and the RNA will be made, the RNA also has both these coding and non-coding sequences. Now, here where the splicing has to take place, where all these non-coding sequences has to be cut out. And then finally, we have the mature mRNA, which then codes the protein. So this only has the coding sequences. Now, in our normal cells, in eukaryotic cells, this is done by the spliceosome. This is a huge, complex, very complicated process, but it does it perfectly, and indeed we end up with a coding message. Now, if the splicing doesn't take place, then this kind of pre-mRNA is just not usable for the cell. It will not understand. So I just put this little example here, and I think you can appreciate that if you have a message, which is in this case the red, interrupted by these non-coding kind of letters, then you don't really understand the message. But as soon as you splice out these non-coding parts, then suddenly you have an mRNA which is only the coding letters are there. And finally, the translation, which will kind of translate this coding to a protein language, which in this case, the dog ran and ate the hat. So this is in general splicing. So we're using this concept, but we are using in a simplified version, which is we don't need the spliceosome for this. We are using self-splicing introns. So what are self-splicing introns? These are found in bacteria and in certain fungi. So normally it's not part of the human body. But the good thing is that these are just simple RNA fragments, which just can fold up in a very specific way. And once they folded, then they can just splice themselves out. And then suddenly you will have only the coding sequence. Now, how can we use this in this synthetic biology approach? So the first what we showed, and actually this was already showed before us, but we indeed confirmed, and it's working perfectly. If you take such a group 1 intron, and we just cleave it at a certain point, so we just make it, split it into 2 parts. So in this case, we have the first part of the message, and then one part of this intron, which is alone inactive. It just doesn't do anything. It can't fold up in the correct conformation that it splices. And we have a second RNA, which again has the second part of this intron together with the second part of the message. Now again, this is just, it's for the cell, this is something gibberish. It would not understand because it has all these non-coding parts. But if we put a guide RNA on both of these, which just find each other, then suddenly this intron can assemble, and again, the splicing can take place. So suddenly we'll have an RNA, which is consisting of exon 1 and exon 2. And the intron is just cleaved out. So this is the basic mechanism that we decided to use, because if you think about this, is an amazing synthetic biology tool, because you can hide a message in this exon 2. The exon 2 is just not understandable for the cell because it would want to start to translate from here, and then it reaches this non-coding part, and afterwards it just gives up. It doesn't go further. So whatever is here is just hidden from the cell. It doesn't make protein, it's just an RNA. However, if we target this together, and in this figure, I just target it together just unconditionally. So this will always find each other and it will splice it out. And so it will be a message. But how could we make this dependent on a specific transcript? And the way how we can achieve this, to add specific guide RNAs. So as you see, we have here the one half of this type of intron and the other. But now we added specific guide RNAs, which can find our target transcript. What is our target transcript? A transcript, for example, what is in patient A is specific for cancer cells, but can't be found in non-cancer cells. Now, as you see, these entire type 1 intron will only assemble to a functional intron if this target transcript is found, and it can, both of these parts finds this target transcript, it assembles, and then the splicing will take place, and suddenly we have this message, which will be expressed in these cells, but only if these cells have this target transcript. In a non-target transcript, so in here we have cells without the target transcript. So here, this is another transcript. It can't be found. The base pairing doesn't take place. So these two parts of the introns, they just never really find each other. And because of this, the splicing will not take place, and all the information that is here is hidden from the cell. So it will be not expressed. The protein just won't be made. And I will not show you now data and gels, but the basic concept is working. So we can now put these constructs into mammalian cells, and we can have specific guide sequence which targeting very specific sequences, and indeed we can see then this splicing takes place, but in a cell where we don't have that transcript, or where the guide RNA is just like a mixed sequence, then this doesn't take place. So it's working. However, we are still working on the efficiency because it's still not very efficient, and we have to optimize further. I just show one more slide, how we can optimize, or just to make you understand that I just showed group 1 introns, but actually group 1 introns are many thousands and tens of thousands, because in various bacteria and various fungi, they all develop their own little type 1 introns, which all look a little bit different and have some specific features. Now, interestingly, these are barely characterized. So almost all work which was done with one specific type 1 intron, but nature has like a huge untapped resources of these group 1 introns. And indeed, we started to screen some, and we realized, oh, some are much more efficient than others, and much more efficient what was already characterized before. Plus, we are optimizing. So we have to do very specific designs, how we cut these introns and how we make exactly these guide RNAs. So without going further into details, but that's exactly what we are developing now. So the basic proof of concept is working, but now we have to make a tool which will be efficient enough to be able to use this in actually cancer cells, targeting cancer cells. And so this is my last slide, and I just want to kind of again refresh. So what our tool can do, we can program it with specific sequences. So these are those little guide RNAs what we can add, and these specific sequences will screen the RNA, the transcriptome in a cell, and if this specific sequence with that mutation, for example, that cancer mutation is found, then the executor program means that then suddenly the splicing will take place, and this protein will be translated. And that protein can be anything. We can choose what kind of protein we want to do. We want to put a cytotoxic protein or some other protein, and this is what Marian will speak about further. So what is our payload? We can decide. But in a cell where the scanning doesn't find that specific sequence, the splicing will not take place, and that payload will be hidden, and it will be not expressed in the cell. And so now I pass back to Marian, how we can optimize the payload. And for that, I need to stop sharing.
Okay, so thank you, Tamas. So so as Tamas just described, so what we have is a potential therapy where you can deliver it as an mRNA, but essentially without the presence of the specific target sequence in the cancer cell, splicing and therefore translation of the protein won't happen. Whereas we can ensure that the expression of the protein then only happens in the cells with that specific mutation. So then the question is, well, what would we want to express? So in the simplest idea, you could just express something which is toxic and so will kill the cell. Sorry, something's, that's it. Yes, so in its simplest form, for example, and I've just listed a couple of examples here, but there's a huge range of different things that you could consider here. But for example, if you expressed a version of caspase-3, which is constitutively active, that would trigger apoptosis of the target cells that had that specific mutation. Or, for example, you could express a prodrug activating enzyme. So then on delivery of the prodrug, only in the cells that are actually expressing that enzyme would you get conversion of that into the active form of drug, which would kill the cells. However, with any of these therapies, it's not going to be possible to target all of the cancer cells. And the reason for that is, firstly, tumors are heterogeneous, so not every cell might have your target mutation, although, of course, in theory, you could design multiple of these targets to target a range of different mutations. However, even if you were able to target all the mutations, it's not going to be 100% efficient in getting translation of your target payload protein. So then the other factor that we've considered is, because the beauty of this system is that you could engineer it to express anything that you like. And one of the key areas of interest in my lab is trying to develop ways for how you might turn a cold tumor hot. So in conjunction with expressing something which directly kills the tumor cells, which will then also lead to release of tumor antigens, which can help prime an effective antigen specific T cell response. We're also investigating expressing other proteins which would then also activate the immune system. And there's a range of different approaches, again, that you could use, for example, expressing cytokines or chemokines, such as IL-12 or CXCL9 and 10, which would lead to T cell recruitment and also activation. Or you could also consider expressing antibodies. For instance, you could express single-chain bispecific antigens that would be secreted in the tumor environment and then could bind a antigen on the surface of the tumor cells, and also CD3 present on T cells, and so facilitate T cell recruitment and also targeting of bystander cells. And by that, I mean cells that are not directly targeted by the therapy itself. So we're currently working on a range of different approaches, and we can test these in parallel with developing the tool itself to work out what would be the most effective combination to both kill tumor cells and also prime the immune response. And we do that. We have a range of different tumor cell models that we use in the lab, so also human cancer cell lines and also mouse tumor models. So just to summarize how this would work, you would start off with an immune cold solid tumor. You would then deliver the mRNA therapy, which in cells which had the specific mutation that it was targeting, that would kill both direct killing of the tumor cells, so the cells would lyse, releasing a source of antigens, which can then be taken up by dendritic cells in the tumor microenvironment to present them as T cells. At the same time, those cells which have been targeted would also secrete locally into the tumor microenvironment immune activating molecules, for example, cytokines and chemokines, which will then facilitate recruitment of T cells, CD8 T cells, which can then specifically target any residual tumor cells left that have not been directly targeted and killed by the therapy. And so in this way, it would be possible that, as well as directly targeting cancer cells, to also turn a cold tumor hot and prime an immune response, which would also take out neighboring tumor cells. I'll finish there, and I just wanted to highlight that this, as we mentioned, is a collaborative project. So we are looking for potential PhD students to work on this project, and this would be supervised in collaboration between the two laboratories. I've listed our lab website here, and you can also find Tamash's lab webpage on the John Curtin website. And there are details on there on how you can apply. And with that, I'd like to finish, and we're happy to take any questions.
Thank you so much, Professor, for that insightful presentation. If you have any questions, as I mentioned earlier, you can obviously use the Q&A button. I'll give a few minutes for our participants to type in the questions if there is any.
Probably just taking just a little of this time. There is, of course, now a lot of hype about mRNA therapies, and probably you all know that now we are using mRNA, directly delivering mRNA to make antigens and kind of for immunizing, like in COVID-19. But now there is a big push to kind of use mRNA therapy for other settings, so not just for immunizing. One of the big advantages with mRNA that it doesn't incorporate into the genome, so you don't have all the problem that, like with DNA editing, that it might edit certain parts of the genome which you don't want, or in certain cells. So overall, developing therapies which are only based on mRNA would have a lot of advantage. One of them, that RNA itself is not immunogenic, or just that's a bit more complicated, but in general, the RNA is the same like our own RNA. So it's not like a bacterial protein will be always immunogenic, while RNA is just one more RNA in the big soup of RNAs in our cells. So we are really developing this as a mRNA therapy, as Marian explained at the end. I'm not sure if there is any questions yet.
We have a question. There's a question on what is the efficiency of splicing itself?
Okay, that's probably I can. So I don't know whether I understand the question correctly. So in general, sorry, I might not directly answer your question, but in general, in our cells, the efficiency of splicing is very, very high. Also, it's close to 100%. Now, but probably your question was more about what is the efficiency of this type 1 intron splicing. So in general, it's about if you just insert an intron, a type 1 intron into a messenger RNA, then right now we estimate that it's about 50%. So 50% of these transcripts are spliced. And the point is that when we cleave this, so when in our system, the efficiency is still lower, because of course we split these introns, and they have to find each other, and they have to find the target, which is right now in the low, so few percentage of the splicing efficiency. So that's not good enough. And this is exactly what we are working to push this into the higher range.
Thank you. Hope that answered your question. If not, please use the question button to ask or clarify any of the questions. We have another question. The question is, how does the prodrug therapy work in the context of RNA?
Marian, do you want to answer, or should I try to answer?
Well, yeah, I mean, I think either of us could answer. I mean, I suppose just to clarify that we're talking about that this would then be expressed as a protein. And so what we are saying is that you could express for example, an enzyme that would then activate the prodrug that would be delivered. So the prodrug itself would not be delivered as an mRNA. It would be the mRNA that was delivered to then splice and express the protein. So yeah, does that answer the question? I don't know if you want to add anything else to that, Tomasz.
Yeah, no, I think it's, yeah. So it means that the prodrug, because prodrugs have to be activated by the prodrug activating enzyme. So in this case, that's the protein which would be translated in the cell. The prodrug would be added just systemically as normal. Did we answer the question? Hopefully.
Thank you so much. Another question. Can we have continuous expression of enzyme?
So no, this is all transient. So when I said that mRNA therapies have a lot of advantages, you can also say that some of these can be also some disadvantages. So of course, anything that you use an mRNA, that will be always transient. It will be in those cells for a couple of days, max a week, but then they will be just degraded and it will be completely disappearing. Unlike if you do genome editing, which will then stay there. So in a cancer context, of course, this is what we want, because we would like to eliminate these cancer cells, and probably would give like repeated treatments. But at the end, when we stop these treatments, we would want that this is gone. Now, for this whole concept could be used also not just for cancer treatment, but for other mRNA therapies, where you would actually want to maintain a longer term expression of the protein. And I will not go into that, but we can operate with circular RNAs, which really extend the time when this protein can be expressed, but it will be, at the end, it will be always transient. It will be never, yeah, it's not like a genome editing.
Thank you. We do not have any other questions, so give it a quick few seconds to see if a last minute question comes through. If not, we will end the session for today. Before we finish, if Professor Marian, if you could put up the last slide of your website, so if there's any participants who would like to take notes of the website, if there's any interest in PhD applications. Obviously, we also have our email address, which I mentioned, which is outreach.chm@anu.edu.au. So if you have any questions for our panelists about PhD at ANU, please do send us your questions and we will make sure to forward them to our presenters today. We do not have any other questions, so I will finish our session, but a huge thank you to Associate Professor Thomas Fisher and Associate Professor Marian Burr for your time and expertise in this absolutely amazing presentation. And I, as a non-science background, learned so much. So thank you so much for this wonderful presentation and taking time. And also thank you for our participants for staying along and being interactive with our session. Thank you, everyone.
Thank you.